Well, I'm just going to say welcome to those of you who have joined us, and I'm going to give maybe just another minute for all of the attendees who have registered and are logging in right now to join. But in the meantime, thank you for joining and welcome. One of the, I think, challenges with this remote working environment, especially using Zoom and all that, is that people have meetings that run up against the top of the hour. And so, you know, they're toggling out of one system into another. And I read something in Blood, I think it was Steensma, who wrote an editorial on the challenges of being on Zoom all day, back to back to back. So he made a recommendation that we should all agree to either start 10 minutes after the hour or end 10 minutes before the hour, so you could have at least some time. I thought it was a pretty good suggestion.
I had a meeting like that, and it used to start at 12 o'clock, and now it starts somewhere after 12:15.
Oh, wow.
And the people still are straggling in.
They're still coming.
But they reset their clock to the time they know the meeting will start.
Right. All right. Well, it is 3:03, and I want to make sure that we have plenty of time for Q&A, question and answer. So I will start the introductions. Welcome, good afternoon, good evening, depending on where you're joining us from. This is the RUNX1 Research Program hosted webinar series. And for those of you who don't know who we are, we're the RUNX1 Research Program. We are a not-for-profit organization. I'm Katrine Erickson, I'm the executive director. We do our work through raising awareness about this rare disease, through patient community building and research community building as well, and of course through research funding. And the funds that we use to stimulate research are really aimed at discovering interventions to prevent the onset of blood cancers in our patient population. We are grateful to our co-funding partners for our research initiatives, and those two organizations include Alex's Lemonade Stand Foundation, as well as the Leukemia Lymphoma Society. We'll continue to host these webinars until we achieve our mission. So please continue to join us. Look out for our emails, which will let you know when there's a new upcoming webinar. Today, as usual, because this is a research webinar, we have mostly scientists online, but you may know that we are making a concerted effort to include our patient community on these research webinars as well. We do this because we think it's extremely important to include patients as partners in research and not just subjects. And so I just want to thank the patients who have joined us today and welcome them as well. As a quick reminder, and perhaps not too surprising, the structure of our webinar is common in that we're going to start with the presentation by our expert scientists today, and we'll end with the Q&A. Hopefully we'll have about 10 to 15 minutes to address question and answers. One other important housekeeping note is to please include your questions or ask your questions through the Q&A icon. I will use that Q&A icon to facilitate the Q&A at the end. Refrain from using the chat function because it's a little bit distracting and it's not as easy to organize the questions at the end. So with that, I'd like to introduce Dr. Morty Poncz, who is a funded RUNX1 research program and ALSF researcher. He is the chief of the Division of Hematology at Children's Hospital of Philadelphia, and he is an expert on congenital platelet disorders, most especially RUNX1-FPD. He's also been an advisor to us since the inception of our organization, and a dear friend, and we're grateful and thankful for his research efforts on discovering interventions for leukemic progression in RUNX1-FPD. And now I'll turn it over to Dr. Poncz, who will introduce his very talented postdoc. Morty?
So thank you, Katrine, and you're right. The major part of this presentation is by a very talented and dedicated postdoc, and Brian Estevez. And what we're going to do today is to discuss research which was mostly supported by this RUNX1 research program. And it's our approach for trying to screen for drugs for the prevention of disease progression in patients with RUNX1-FPD. And what I will do is set the stage of why we use the approach we had, what we accomplished over the past 2 years of support, and end with a discussion of where we want to go forward. So come next slide. Thanks, Brian. So the underlying idea is that we believe that the mechanism leading to the increased risk of AML and RUNX1 haploinsufficiency also underlies the platelet defects. The implication of this is that correction of the platelet defect will also correct the tendency to have an increased risk of AML. Now, why did I pick this? I picked this idea or what we call hypothesis out because it plays to my laboratory strength. But also, the platelet defect that's present, while it isn't the major goal of the funding agency, it is still has a very significant impact on the lives of many of the patients that have RUNX1 haploinsufficiency. And this is something that virtually all patients have, even those who may never develop the leukemic process. The other thing is that platelet counts are easy to measure, and the correction of the platelet defect can be followed both in animal models as well as in future human studies, so that if one uses it as a readout of success in therapeutic intervention, one can, I think, approach the problem in a much more rapid fashion than if one is looking for the oncogenic propensity that underlies the increased risk of having AML develop in patients. And as I mentioned, we have a long history of doing research on megakaryocyte platelets. We have a lot of tools in this area, and we have a lot of experience with thinking these through and using the right tools to address the problem. And what we're hoping is that in the end we'll have a model that can be used to look at RUNX1 platelet defects, but also that could be converted into a model that can test oncogenic propensity, as you'll see at the end of Brian's presentation. Come next slide. Thanks. So when Brian began his work, we were using iPS cell-derived megakaryocytes. So these are cells, the iPS cells, which are adult-derived embryonic-like cells that can be differentiated into megakaryocytes. And like other laboratories, in both published and unpublished data, we could reproduce the defect in the development of megakaryocytes, which in that middle part of the figure with the red arrow is an absence of megakaryocytes in the affected line, which is present in the wild type on the left. And then having established this, we went ahead and set up with facilities at the University of Pennsylvania for screening, drugs that are in clinical practice already or potentially could come to clinical practice. So we'll present that data, but at the same time too, we became uneasy about doing these experiments because, as I said, these are embryonic-derived megakaryocytes. And megakaryocytes from an embryo, that is the first few weeks of life after an egg is fertilized, has features very different from megakaryocytes in adult patients, and the entire hematopoietic process is different, and it goes through changes, major changes, as you go through and mature. And as we know, this disease that we're interested in affects adults and adult hematopoiesis. So under number 2 here, we began to use adult marrow that had been donated by healthy controls and recreating the disease in them. And recreating the RUNX1 haploinsufficiency, that is lower levels of RUNX1, allowed us to reproduce the megakaryocyte defect again. And I, just for artistic sake, redrew the figure, but basically the same degree of low platelet yield in culture was the same. And we were able to go ahead and rescreen the drugs that we had done in the iPS cells, and Brian will present that. And then the next step, which is a step where Brian has taken the lead on, is to think of how we could do this in vivo. These studies are in vitro, that is, they're in culture conditions and not in a living organism. And Brian decided that the next step that we should try is to put the marrow, the human marrow, after you convert them into RUNX1 haploinsufficient marrow, back into mice, and that we would study them. So the facilities at at the University of Pennsylvania include a number of investigators that have used this technique a lot. And we have used it a bit, this idea of xenotransplantation. And so our future studies is to put the RUNX1 marrow back in, to document that the platelet counts are low, then to do drug screens in them of the drugs that we studied in culture, and then also to think about how we could use these mice to study oncogenic propensity. And then finally, we think that if others or our own studies suggest that a particular drug or approach might be useful for therapeutic intervention, we propose that before human trials are to be done, which are extremely expensive and take a long time to accomplish, that we go and screen those drugs in the murine models that we're setting up in order to decide when a commitment should be made. So with that as a background, I'm going to turn the stage over to Brian.
Okay, thanks for the introduction, Morty, and thank you to the organizers for this invitation to present our work. So just some brief outline of my talk. So first, I'm going to start by giving some background on the biology of RUNX1, megakaryocytes, and platelets, and something that's less understood, which is that the disturbance in megakaryocytes affects stem cell biology. And then the second part of my talk is going to discuss the drug screening studies. So particularly studies focused on iPSC-derived cells, studies on CD34-derived cells that were confirming those studies, and as Morty mentioned, future plans for bone marrow transplantation of human adult CD34-derived cells into mice. So as many people in this audience are aware, RUNX1 is a transcription factor critical to hematopoiesis and often linked to myeloid leukemias. And RUNX1 insufficiency manifests itself as a quantitative and qualitative inherited platelet disorder associated with an increased risk of leukemia, and here we'll call it RUNX1-FPD. So the goal of these studies is to correct the megakaryocyte defect in RUNX1 insufficiency. So as many of you may not be aware, some of the terms I'll be discussing, I will just briefly outline that we will be discussing various blood cell types, such as hematopoietic stem cells, which are immature blood cells that can develop into all types of blood cells. Megakaryocytes, which are large cells in the bone marrow that produce platelets. And platelets are anucleate cells important in bleeding, thrombosis, and aspects of immunity. So just to give an overview, hematopoietic stem cells are found in the bone marrow. These are primitive cells that commit to the megakaryocyte lineage, and in doing so, they form a cell called a MEP, or a megakaryocyte erythroid progenitor, which then goes on to form megakaryocyte progenitors. These megakaryocyte progenitors continue to divide and make megakaryocytes. And now megakaryocytes undergo a complex maturation process that has several steps and has been well characterized, including the formation of these long membrane extensions called proplatelets. And those proplatelets are what's released into the bloodstream to produce terminal platelets. So these collective processes that I just discussed are known as megakaryopoiesis and thrombopoiesis. So megakaryopoiesis describes the differentiation and maturation steps for megakaryocyte production from stem cells, whereas thrombopoiesis describes the megakaryocyte membrane extension and shedding processes by which platelets enter the bloodstream. And as many of you are aware, platelets are important for stopping bleeding. The RUNX1-FPD phenotypes that are well described are mild thrombocytopenia, bleeding, eczema, and elevated risk of myelodysplastic syndrome and acute leukemia. So the biological mechanisms behind the mild thrombocytopenia and bleeding are not well understood, but have been attributed to dysfunctional megakaryocytes and platelets. And similarly, the elevated risk of myelodysplastic syndrome and progression to leukemia is also not well understood, but has been attributed to early onset clonal hematopoiesis. And in addition, it's unknown if there are links between the dysfunctional megakaryocytes and platelets and early onset clonal hematopoiesis, in particular, whether there are pathways related to the two defects. So the role of RUNX1 insufficiency in mature megakaryocyte development and platelet release has been studied, and it's been shown to cooperate with various transcription factors during megakaryocyte maturation to promote the process, including the release of platelets into the bloodstream. However, the role of RUNX1 insufficiency in stem cells and progenitor cells is not well understood. So I'm going to introduce some studies that highlight the importance of megakaryocytes, which in this study was shown by an inducible megakaryocyte depletion model. So in this study, they used an inducible megakaryocyte depletion model that marked stem cells that express von Willebrand factor, which is a megakaryocyte-associated protein. So those cells are shown here in these pink circles. And these cells have the special properties that they're biased toward making other cells of the myeloid lineage. And what these investigators found is that in the bone marrow in mice, these megakaryocyte myeloid biased stem cells are found physically beside the megakaryocytes in the marrow. And what they did was they induced megakaryocyte depletion in these mice and found that when megakaryocytes were depleted, you had expansion of these myeloid biased stem cells. And in addition, there was functional defects in the stem cells. So to describe this study, what they did was they used these triple transgenic mice, which express von Willebrand factor reporter, GFP. As shown here in this immunostaining, you can see that the VWF expressing cells in the mouse included megakaryocytes, which are these large green cells shown here, but also included these smaller stem cells in the marrow. And what they did with these mice was then, in this background of this von Willebrand factor transgenic mouse, induced megakaryocyte depletion upon exposure to diphtheria toxin. And what they found was quite remarkable, which is that when you deplete megakaryocytes from the marrow, the myeloid bias VWF expressing stem cells are dramatically expanded in these mice. And they also found in studies that I'm not going to have time to go over is when you induce megakaryocyte depletion, you also cause functional defects in the stem cells, as they showed by transplantation studies. So another interesting finding was by another group where they showed that in mice exposed to various forms of myelotoxic stress, here shown as 5-fluorouracil, a chemotherapeutic, when mice are given a chemotherapeutic, it kills the rapidly dividing cells in the marrow. And they also compared this myelotoxic stress model to a different model of abnormal myelopoiesis in the BCR-ABL leukemia mouse model. And in both these studies, they were interested in the effects of megakaryocyte depletion on myelopoiesis. So what they found in the 5-fluorouracil myelotoxic stress model was that they could find a consistent expansion of granulocyte progenitors after 12 days of 5-fluorouracil exposure in the marrow, as shown by these violet cells that would expand predictably by between 10 and 12 days and then differentiate into mature cells, which is consistent with a stress myelopoiesis response in the marrow of normal mice. So what they decided to do was deplete the megakaryocytes in this model of 5-fluorouracil induced myelotoxic stress, and then see what happens to the myelopoiesis. So I'll just walk you through this. On the 4 left panels, they're looking at a later time point, which by this time point, the granulocyte expansion that they observed should be resolved, and you should only see mature blood cells. So what they saw in the controls was that they saw some mature blood cells shown in green, and also some megakaryocytes in red by days 16 and 18 after myelotoxic stress. But upon megakaryocyte depletion, you can see in the bottom 2 sets of panels that there was still granulocyte progenitor expansion that would not reside, suggesting that megakaryocytes suppress abnormal myelopoiesis upon exposure to stress. And what was interesting was that in the same study, using a different model of BCR-ABL induced leukemia, they found a similar result where megakaryocyte depletion caused abnormal myelopoiesis to persist in these mice. So we hypothesized, based on these studies and our own data, that megakaryocytes regulate stem cell expansion functionality under steady state stress and leukemic conditions. And so we proposed that drugs that correct megakaryocyte differentiation defects in RUNX1 insufficiency will also correct dysfunctional megakaryopoiesis and bleeding, and suppress the early onset clonal hematopoiesis and progression to MDS and AML. So we use an induced pluripotent stem cell model to derive hematopoietic progenitors and megakaryocytes in order to study RUNX1 insufficiency. And there's several advantages, as Morty went over, in that they enable the study of mechanisms without repeated blood donations from donors. And also they allow stable genetic manipulations to be made in hematopoietic progenitors, megakaryocytes, and platelets and other blood lineages. But they have limitations in that iPS-derived HPCs, megakaryocytes, and platelets, as Morty described, are embryonic and have reduced functionality. And they may not mimic all aspects of the disease biology. So we use 3 iPSC lines in this study, the most important of which is shown here in the middle in red. It was derived from a RUNX1 insufficiency patient called line 1, which we call L1, which harbors a exon 4 splice site mutation in RUNX1. And this line was corrected by CRISPR-Cas9 to make a paired control called L1 corrected. In addition, we have a wild type line where the RUNX1 exon 4 splice mutation was introduced into a wild type background called wild type L1 and its paired control, wild type. And we also, in some studies, looked at a different RUNX1 insufficiency mutation, which had a mutation in the transactivation domain of RUNX1. And this line is called L2, or line 2. And we compared it to its corrected control called L2 corrected. So to generate hematopoietic progenitors and megakaryocytes, we used evaluated or previously published differentiation protocol to generate non-adherent or floating hematopoietic progenitors after 7 days that we could then collect and differentiate into megakaryocyte after 5 days. So using this approach, we found that, as expected, all RUNX1 insufficient lines showed a defect in megakaryocyte yield, as shown here as the number of megakaryocytes produced per input hematopoietic progenitor cell. And these results are important because they confirm previously published studies with iPS cells from patients, as well as studies from primary cells from the bone marrow of patients. So having established that the RUNX1 insufficiency iPSCs have a megakaryocyte defect, we then screen drugs for effect on megakaryocyte yield. So we also searched the literature for drugs that affect RUNX1 activity and then performed the general screen. So our lead candidate drug is Repsox, and Repsox is a small molecule competitive inhibitor of TGF-beta receptor 1 kinase activity. And it's been shown previously in the literature to promote megakaryopoiesis in human megakaryocytes and increase platelet counts in mice and increase RUNX1 levels. And just to show that in that report, when they looked at hematopoietic gene expression in Repsox-exposed CD34-derived megakaryocytes, they found that RUNX1 was among the genes upregulated. So when we tested Repsox, just to give you a background, TGF-beta 1 acts by, TGF-beta 1 acts by binding to the TGF-beta 1 receptor and activating a signaling pathway that in the end regulates cell proliferation and stem cell quiescence. So this is where Repsox acts upon by inhibiting this pathway. So with this in mind, we used Repsox to treat RUNX1 insufficient megakaryocyte cultures, and we found that Repsox improved the yield of megakaryocyte cultures from our RUNX1 insufficient lines. So we were interested in this, and we hypothesized initially that this was due to elevated TGF-beta levels in RUNX1 insufficient megakaryocytes. However, we didn't find an elevation in TGF-beta 1 levels in RUNX1 insufficient megakaryocytes. So then we thought that perhaps that the signaling was actually enhanced in those megakaryocytes. So to test this, we cultured megakaryocytes with increasing doses of TGF-beta 1, and we found that, in fact, whereas control cells go down to 50% of initial baseline yield for megakaryocytes upon exposure to the TGF-beta, the RUNX1 insufficient cells go down to 10%. And in addition, at lower doses, the megakaryocyte yield was further suppressed compared to controls, suggesting that they are more sensitive to TGF-beta exposure. So to better understand the biochemical pathway underlying this defect, we looked at the classical TGF-beta 1 pathway. And however, we didn't find any elevation, any significant elevation in any of the members of the TGF-beta pathway. However, when we looked at a non-canonical member of the TGF-beta pathway, which is c-Jun kinase, we saw that c-Jun activity, which was elevated in RUNX1 insufficiency by immunoblotting. And c-Jun kinase is interesting because c-Jun is known to be regulated by TGF-beta 1 as well as cellular stress and inflammation. So we further pursued c-Jun kinase by using c-Jun kinase inhibitors such as JIN8 and J9, JIN8 being an irreversible inhibitor and J9 being a reversible inhibitor. So what we found was that in megakaryocytes culture exposed to JIN8, we saw an improvement in megakaryocyte differentiation as shown by these flow cytometry plots of cultured megakaryocytes. And we also saw an improvement in megakaryocyte yield in megakaryocyte cultures exposed to JIN8. So with this background, we pursued drug screening in these RUNX1 insufficient patient-derived megakaryocytes. And so to do this, we seeded hematopoietic progenitor cells from RUNX1 insufficient iPSCs, and we induced megakaryocyte differentiation with TPO and stem cell factor. And then we fixed and stained the megakaryocytes and stained for CD41, CD42B, and DNA to mark healthy, mature megakaryocytes, and quantified the results by high throughput image analysis. And in this study, we looked at 3,000 compounds, and over 100 were FDA approved. Sorry, over 1,000 were FDA approved. So shown here are some representative results where we show the candidate compounds. In the blue circles are the compounds that met the statistical cutoff for significant effect compared to vehicle control. And so we had several compounds in hand that met the criteria for improving megakaryocyte differentiation defects. So with these results, we decided to validate a subset of these compounds in iPSC-derived megakaryocytes. And we found that indeed some of these compounds did improve megakaryocyte differentiation from RUNX1 insufficient iPSCs. However, as Morty mentioned, the iPSC-derived megakaryocytes and the system in general is embryonic in nature, having several significant differences. So we pursued a verification in an adult human model of RUNX1 insufficiency. So this has several advantages. It enables the study of disease mechanisms affecting adult human HPC megakaryocytes and platelets. And as was mentioned, RUNX1-FPD is an adult disease. So these cells are not likely to harbor secondary mutations, and they're open to stable genetic manipulation. So the limitation is that they have a short lifespan and experimental window for use in culture. So with this system, we introduced RUNX1 insufficiency by lentiviral shRNA transduction of CD34 hematopoietic progenitor cells. We then sorted these cells for GFP and cultured them in megakaryocyte culture conditions, and then exposed them to drugs. So to verify the system, we optimized the conditions for knockdown, and then we looked at RUNX1 mRNA levels. And we found that using two different shRNA vectors, or short hairpin RNA vectors targeting RUNX1 gene expression, we saw RUNX1 expression was reduced to 50% of control levels, and about that in using a different shRNA. In addition, and consistent with these results, we saw a significant reduction in megakaryocyte yield using both shRNAs targeting RUNX1. And these results are important because they show that we can introduce RUNX1 insufficiency in an adult model of hematopoiesis. So we also decided to further characterize these cells and the effects of RUNX1 knockdown. So when we looked at colony assays for multilineage growth, we saw that as expected, the megakaryocyte colonies were inhibited by RUNX1 insufficiency. And we also saw a moderate effect on erythroid colonies and no effect on granulocyte macrophage colonies. So going back to the screen, we induced RUNX1 insufficiency, as I mentioned, and then this time we looked at the top hit drugs from the iPSC system for effects on megakaryocyte yield. And when we did this, only some of the compounds had some effect on megakaryocyte yield in the adult system of RUNX1 insufficiency. So we further validated these studies. This time, our comparison was against a drug that's a TGF-beta receptor 1 inhibitor under clinical development called gallinesertib, abbreviated here GS. And we found that TGF-beta receptor 1 inhibitors and TGF-beta receptor 1 associated pathway inhibitors significantly improved megakaryocyte yield in this model. And the effect of Repsox, which I don't show here, was selective to the megakaryocyte system, and it didn't affect myeloid lineage or erythrocytes. So the next thing I want to talk about is toward establishing an adult humanized mouse model in order to validate some of these studies and pursue compounds for in vivo effects. So the advantages of an in vivo humanized mouse model is that it enables study of human bone marrow hematopoiesis and platelet functionality. And following up lead drugs for effects on long-term functionality and myeloid and platelet levels is actually very important, especially in this disease. And another thing that it provides is insights into clonal hematopoiesis. So the limitations of this model are that it may not mimic all the disease-related changes, such as effects on the niche of the bone marrow. So in this model, we propose to induce RUNX1 insufficiency, as I mentioned before. Now, this time we will take the RUNX1 insufficient progenitor, CD34 positive progenitor cells, and inject them into these MBSGW mice, which are immunodeficient mice that have that they harbor a KIT mutation, which is a stem cell factor receptor, allowing them to engraft hematopoietic cells more easily than traditional immunodeficient mice. And with these mice, we will bleed the mice regularly to assay for peripheral platelets, leukocytes, and myeloid cells, as well as isolate the bone marrow in order to see the effect of RUNX1 insufficiency, particularly focused on the effects on platelet counts for screening drugs. So as I mentioned, this model has an interesting ability, is that it can allow the study of clonal hematopoiesis, which it can be done by performing competitive transplantation studies with these mice. So what we envision is that using the RUNX1 insufficient GFP expressing hematopoietic progenitor cells, and as well as the GFP negative transduced hematopoietic progenitor cells, one can mix these in predetermined ratios and then transplant them into these MBSGW mice. And by using lower input numbers of GFP positive cells than the GFP negative cells, one can see if that over time, the RUNX1 insufficient progenitor cells acquire a growth advantage as indicated by an increase in GFP positive cells in the peripheral blood over time. And if this is the case, then it will be easy to design a therapeutic strategy, whether intervening before clonal expansion or after clonal expansion to deliver drugs for screening into mice. So as I mentioned, we started to summarize and conclude, we started RUNX1 insufficient drug screening in iPSCs. And now we've developed an adult lentiviral CD34 system that recapitulates the iPSC system. We now propose incorporating the adult system into the xenotransplantation studies in mice, as I just described. So the xenotransplanted mice are useful for further drug screening and confirmation of effects on megakaryopoiesis, platelet biology, and leukemic evolution. And they also may be useful as a preclinical method to save time and money. So I'd like to acknowledge my advisor, Morty Poncz, and the Poncz Lab, all the collaborators at CHOP and UPenn for their extensive scientific and technical expertise in this area, and also the RUNX1 Research Foundation for their tremendous support here, as well as the patients and the families for their interest and enthusiasm. Thank you.
Great, wonderful. Thank you, Brian. Thank you, Morty. Really appreciate you taking the time to share your data with everyone. We do have a few questions, and unless you had any other comments you wanted to add, I will get started on the questions.
Sure.
All right.
Sure.
So the first question is, what is the effect of REP-SOX on hematopoietic stem cell quiescence?
So I don't think this has been directly tested, Although presumably, because it's a TGF-beta receptor inhibitor, it would release cells from quiescence and actually induce expansion. But that's in the normal setting. We don't know in the context of RUNX1 insufficiency if this is the case. And in fact, no one's actually done those experiments.
Thank you. There's a question from a patient with regard to biologics such as Enbrel and Humira, which are TNF alpha inhibitors, and they're used for the treatment of RA symptoms. Is there any data to suggest that they would have a negative effect on RUNX1 mutation carriers, causing them to transition potentially to frank leukemia, is the question.
I'm not sure about those studies, and I really couldn't comment. I'm just not sure.
What I would say is that we should test them in our system and see what they're doing. And certainly we can try them out in the murine system when it's up and running.
Yeah, they're certainly involved in inflammation and the inflammatory pathways, which you said were elevated in your system. So it could be very interesting.
I'd be happy to test any compounds. If anyone wants to email me some compounds to test, you can add them in. It's not hard.
That's great. Well, thank you for offering. Another question is, well, comment and then a question. Nice work. Have you thought of using the RUNX1 heterozygous mice in the in vivo studies?
Yeah, we've discussed using the het mice. The problem with that, so for sure, it's occurred to us. It's an interesting model, and it does have some resemblance to some other phenotypes. The problem with the mouse model is that it doesn't recapitulate the leukemia predisposition, as the homozygous doesn't either. And so we're afraid that we would be modeling mouse, a disease, things that are not relevant to the human disease. So we're more focused on creating a humanized model of RUNX1 insufficiency, if that makes sense.
Yeah. I'd like to add that these are subtle events. That is, hematopoiesis is almost identical in humans and mice, but there are subtle differences in RUNX1 haploinsufficiency. Remember, you still have a lot of RUNX1 around. It's just half normal, approximately, and you have this devastating outcome. And that devastating outcome is on people, but you don't see it in mice because, probably because of the subtle nature of the disease, the subtle differences between hematopoiesis in the two organisms. And then two, drugs that work in people may or may not also work in mice. So that if you don't find a good outcome with a particular clinically useful drug for people, is it due to some difference between human and mice studies? And so one of the advantages having put human marrow in mice is that a lot of the targets, not all of them, are now human targets. And so you could study a relevant drug for human without worrying about species differences. Is the mouse more sensitive, less sensitive to the drug than humans is a big issue.
There's another question from a patient, which is in regards to taking a medicine called Nplate, and whether, and this particular patient says they did actually get an increase in platelet numbers, and they're curious whether this is something they should consider taking if indeed your hypothesis is correct that rescuing platelet numbers could potentially have an effect on progression to MDS or AML.
Go ahead.
I would say that's a very interesting observation, but that needs to be carefully studied. I think that would be a very good candidate for our preclinical model, for example, but I'm not sure what the long-term consequences would be of being on this drug.
So I would add to that one of the things that we haven't studied yet, and I've been trying to drag Brian kicking and screaming to do, is to look at the function of the platelets that are released and whether the drug not only corrects platelet numbers, but also their dysfunction. So not only is the platelet count lower, but the platelets have multiple different defects in them. And so you may be able to correct the platelet count and still have a bleeding disorder, or you may correct the platelet count alone and not have an effect on the underlying differences like TGF-beta-1 sensitivity may remain, and the inflammatory changes may remain too. In addition, the thrombopoietin mimetics also affects stem cell multiplication, so that if you take a drug and you have a pre-leukemic disposition, are you going to tickle that process as well as tickle megakaryocyte production and platelet count, so that you can get into more trouble rather than eliminate trouble by being on continuous end plates. So I think it's worth studying. I think that and I can't recommend therapeutics, but if you're going to take it prior to a procedure, such as a surgical procedure, it might be useful for short-term exposure without increasing long-term risk of clonal evolution. That is, to be on it prior to the surgery to get your platelet count up and continue platelet count up for a period of time. And I mean weeks after the surgery, so that you can control bleeding better and avoid platelet transfusion. It's a possibility, but I think it would need to be studied. And we certainly, I hadn't thought about, but we certainly could throw it into the mix of things we look at.
Yeah.
Great. Another question I had actually was, if in your system, your xenotransplant system, you don't actually get an expansion, what are some other ways you've thought about testing this linkage in terms of rescuing megakaryopoiesis and affecting the stem cell compartment with an ultimate goal of preventing clonal hematopoiesis? So is there another system or another way in which you can test that?
Yeah, I think, so I think it's been shown that if you continuously sort cells that overexpress a particular RUNX1 mutation in culture for several weeks, they eventually start to expand out abnormally. So that would be a potential strategy. Or another strategy would be to expose the cells to inflammatory stress and see if they gain a proliferative advantage in culture compared to control cells, to look at the effect of stress-induced expansion compared to control cells, whether the progenitors will proliferate better under those kinds of stresses, although we're not sure of what kinds of stressors would induce that kind of response, but that would be one way to start to approach the question in vitro.
Great, thank you. I have no other questions. Are there any other questions from folks online? Well, I think that's all for today then.
Great.
Thank you so much, Morty. Thank you, Brian. Really appreciate it. Wonderful work. We're looking forward to seeing progress in the next update. We'll have you back maybe in a year's time to hear how things have gone.
Thank you. Yeah.
Take care. Bye-bye, everyone.
Bye.